Yes, then I'll share my screen. Yes, so what you should do is you should enter arabidopsis.org. This is the ultimate source for arabidopsis. Here you can find a lot of things. Even I am not aware how much you can do here. So for example, you can download microarray results, public data. You can look for protocols like how to grow arabidopsis and so on. And what's good here is, there are a lot of databases from external sources about arabidopsis, like they did yeast to hybrid experiments and look for interactions, and then they put it somewhere. And another group did some other experiments like microarray of some certain stage of a plant, and then they put it somewhere. And in this website, you don't only have all these things that belongs to this website, but also links to the external sources. That's why I think this is an ultimate source. And here you can even find where you can find arabidopsis mutants. So you need to order after you find out which lines you need. And then you'll go here, and then these are the sources where you can order the arabidopsis lines. How do you start examining your gene or mutant of interest? So you push here and you can do some search. There are many different types of search. I think the most commonly used one is the gene name. For the name of the gene, you have two options. Either you can write the ATG number of the gene. ATG is standard gene name for arabidopsis genes. Or you can write the popular name of the gene. Like for sodium transporters, did we say HKT or AKT?
For potassium, I think it was AKT.
Yeah, sorry, AKT.
Family, if I'm not wrong.
You can directly write AKT here. If there are some similar named genes, yeah, so 13 genes came up. You can read here the descriptions. I think this is a phosphatase, not a transporter. I think you're looking for more transporter. So this is a potassium channel, AKT6, for example. And all these, I mean, this is also AKT1. Do you have a particular AKT you want to check out, or should we just go one of them?
I don't have any particular one, but I talked to Emrah Hoca, and he said for now we are gonna do very simple experiments. So he said don't go into very detail about the.
Yeah, so I'll just show you an example. Like we can take AKT1 as an example, and I'll show you what you can do using this website. So I push here. So here we have all the general information. For example, all the names that you can find in the literature for the same gene. Sometimes two groups find the gene at the same time, and then they give two different names, for example. So you can find them here. And here gives a description, not always very good description. Usually the guys who characterize the gene puts all this information. But it's good, you have a general idea what it does. Here you can see the alternative splices, like this one and this one is, I mean, two versions of the same gene. And here we have Go modules. Do you have any idea what Go modules are? So no. Yeah. So Go things are general categories, and there the reason to have this is when you do a microarray experiment, Just a second. My cat came from the. Okay. People do microarray experiments and they want to examine which pathways are affected by the stress, for example, they put. And then if every gene is put into some categories, then they can say 20% of all the genome is involved in, for example, growth and development. I mean, genes that are related to growth and development, or like 10% of all genome in this stress is affected from, no, maybe it was not a good, sometimes with Go figures, we have like drought stress, for example. They put the gene in this Go figure, Go category, and then they may say after microarray experiments, like 20% of all genome is the genes belong to drought stress, for example. I mean, this kind of stuff. Okay. Then you can find the sequence. Yeah, you can have a look what you have. And then here you have the mutants. You can use this area to find out the mutants, germplasm. And for each mutant, you may have a description of the phenotype. So when you wanna order, you should give this name or this name or this name. But there is another method, which is, I think, better, using an external source. So here are the external links. And tDNA Express is the one which we use to find out the mutants. It's a bit complicated if I don't tell you how to use it, so I'll move to this one. Okay. Yeah, it seems really complicated. So the gene of interest is this one, because it's green, you see, I mean, and these are the other genes. What we can do is to focus using these arrows, then there will be less, it'll be less complicated. Okay, so this is the gene of interest. You can see it starts from here, and this transcript starts from here. So this is the first exon. This is the end. And this is the genome. And here, wherever you see some names, these are the mutants. So for you, the most important mutants are Salk mutants. So these ones, Salk. I'll only describe these Salk mutants. The others for you are not that important, except sometimes if you don't find Salk mutants, you can go for Sale mutants. For Salk mutants, you have two types of mutants. One of them is like a homozygous mutants. You can see it's written HM. You see HM, yeah. This means homozygous. And this one, for example, another line, it's not homozygous. So first thing is to find the homozygous mutant which is in your genes region. Yeah. Okay, you can see they are not like, they are like arrows, and the reason to have arrow is so they use agrobacterium to produce these mutants. Agrobacterium is the bacteria with the tDNA. It randomly inserts the region into the genome of the plant, and then if this thing enters into the gene, then it disrupts the gene. So then scientists do a PCR using flanking regions of this tDNA insertion, and they do a PCR, and this is the PCR result. So what this means is probably insert is inserted somewhere here, and when they did the PCR, you know, the first base pairs you usually don't get, and then they started getting the PCR product and then they overlap it with the genome, so it's somewhere here. What does it mean? If you order this mutant, it's highly likely that at least this one is knocked out, because the tDNA should be somewhere here, and if it is close to the first exon, it's usually a knockout line. But there's possibility that it can be a knockdown line where transcription rate is decreased. And another possibility, I think there must be a promoter of this gene somewhere here, but I'm right now exactly not sure. It looks a bit strange to me. I mean, I don't understand how there is so big intron here. Maybe it's a mistake, I don't know. So maybe this thing hits the promoter region, which controls the gene expression, and then in such a case, you may see like overexpression of the gene, knockout of the gene, or knockdown of the gene, or no difference at all. I mean, everything can happen. Okay, we don't have any other line, I think. We have one here, but I think it hits somewhere here. It probably didn't hit our gene of interest. Can you follow me easily? I don't know how good your genetic background is.
I think I understood
You
what
could.
were you explaining.
Okay. Do you have questions?
For now,
Mhm.
but for example, we are not sure what this gene, this SOC, is doing. Is it knockdown? So we have to first order and do experiment and see what happens. There's no other way.
Yeah, but it's also possible that some others did some experiments using this line. So what we should do is we click on it, and then there will be more information. So here is what comes up if you push it. Usually when it says it's a homozygous knockout line, it's usually really knockout line. So someone probably analyzed it and then published it. And then you can order from ABRC or NASC. There are two big places where you can order. So when you push here, they will give you more details how to order it. And yeah, I don't think you can directly see the publications associated with it. But what you can do right now is to note down the name of the line. Okay, Solk 718 something, and then go back here. Solk 718 something. So this one, okay, this is the one we have seen. Okay, there is no description here. Maybe nobody used it. What you can do now, you can copy and paste the name, this name, and it may hit some literature, but it may not as well. Sometimes you need to put C if it's homozygous. Sometimes they show like this. Okay, you see when I put C, then did you see it, right? I mean, there's an article using this line. So you need to put C if it is homozygous at the end of the name. Maybe it's an important thing to note down. Then you should go to this article. So the authors of this article, I think, didn't put the necessary information here, but yeah, people usually don't put. And you can see there must be another line, but I think it's a special line. Maybe the guys who did the article, who wrote the article, produced it. It's possible because we don't see it here, I think. But you can have a second look. I mean, maybe it's there, but I miss it. Okay, or you can push this and then you can get more information. So it's published in Science. Yeah, so you can look for where to find this. It has a WS2 background. I don't know you have any information about it. So usually we work with Columbia 0 background, COL. Sometimes we don't have these mutants in Columbia background, then you can go for another different backgrounds. But everything will be more difficult if you use a different background, because all these tools are generated in Columbia 0. Of course, there are exceptions to that. Okay, so this is how you find the mutant and then order for your experiment.
How long does it take until they bring the?
I think it doesn't take too much. It takes less than 1 month. I would be surprised if it takes more than 1 month. And I would say it can arrive in 2 weeks. And the prices are quite low. So only $8 per seed usually. But there are exceptions. Sometimes they are expensive. But most of the time, $8. Another thing I like very much, but I don't know, maybe it's not of your interest, so I will just show it as a glance. So we have this EFP browser. It shows us very nicely in a very visual way which part of the plant we see the expression. For example, this is the arabidopsis plant, whole plant, and where we see red color, the expression is a lot like here you see. And in the mature plant, we don't see this expression a lot for this gene. It's usually expressed very much directly after germination. And maybe after germinating, I mean, when it is mature, but in vegetative stage, early stages, we see some in the root also. Yellow means it's almost not expressed. And here we have seeds. Here we have flowers, unopened flowers. Here we have leaves, different stages and so on. Yeah, and then you can see under which stress how the expression is changed. Like this is all derived from microarray data. Yeah,
It's actually very cool.
yeah.
I would like them.
Yeah, and it makes your presentations look professional because they are pictures. Yeah, like very different conditions you can find expression. So this is, for example, where they cut the pieces and then do tissue culture, and how the same gene is expressed under different tissue culture conditions and so on. There are a lot of things you can look for. Like you can focus on seeds. It even shows you the different parts of the seeds, how they respond. I mean, even the arabidopsis seed itself is extremely small. They show you here the cell layers. You see, this is extremely small normally, but you can see the gene is expressed in the seed coat, but not in the embryo. You can differentiate. It's really nice.